Today we'll be discussing regarding KERB cycle, that is our TCA cycle. I'm Shaista from Department of Studies in Microbiology, KSOU. We'll be studying about the KERB cycle, its regulations, and the energetics of the KERB cycle, and how it is linked with various other cycles in today's class. Okay, so let's proceed. Acetyl-CoA is an important precursor of TCA cycle, that is tricarboxylic acid cycle. So let's understand what are the various sources of acetyl-CoA. The first being the carbohydrates. The cell always prefers carbohydrates as the first source. The carbohydrates, that is our polysaccharides, are converted into glucose. Our glucose undergoes glycolysis. We have learned earlier that the 10 steps of glycolysis converts the glucose into a 3-carbon compound pyruvate. The pyruvate now undergoes decarboxylation in the mitochondria to be get converted into acetyl-CoA. Okay. When all the carbohydrates is used up, then the cell shifts its to the next source, that is our fats. Fats on lipolysis gives fatty acids which undergo beta oxidation so that they can be converted into acetyl-CoA. Now our third source for the acetyl-CoA is amino acids. We know various proteins are buildup of various amino acids. The amino acids results in catabolic breakdown. If the amino acid is glucogenic, it is getting converted into pyruvate. If the amino acid is ketogenic, it is getting converted into acetyl-CoA. Thus, a huge reservoir of acetyl-CoA is available due to the various sources like carbohydrates, fats, and proteins, which can be broken down into acetyl-CoA. And this acetyl-CoA can enter a tricarboxylic acid cycle, which is one of the possibility of utilizing this acetyl-CoA. So the citric acid cycle is also known as tricarboxylic acid cycle or the KERB cycle. So it is defined as the cycle which involves the oxidation of acetyl-CoA to carbon dioxide and water. This cycle occurs within the mitochondrial matrix. Even the conversion of the pyruvate to acetyl-CoA also occurs within the mitochondria. The cycle occurs in close proximity to the electron transport chain, which also occurs within the mitochondrial matrix. TCA cycle is also known as KERB cycle. TCA cycle essentially involves the oxidation of acetyl-CoA to carbon dioxide and water molecule. This is the central metabolic pathway. This metabolic pathway is common final oxidative step for most of the molecules like carbohydrates, fats, and amino acids. TCA cycle is an important cycle which supplies energy and also provides many intermediates which are required for the synthesis of various amino acid, glucose, etc. This cycle is an important central pathway which connects almost all other individual metabolic pathways that are occurring within the cell. Okay, it connects various individual metabolic pathways as well. So students, in the earlier classes, we have studied that glucose is converted into pyruvate using various 10 steps, and the resultant product is our pyruvate. We have understood in our earlier class. Okay, the 10th reaction is shown in this diagram. That is, you can see here that phosphoenol pyruvate is converted into pyruvate due to the action of enzyme pyruvate kinase, and where there is a substrate level phosphorylation resulting in a formation of ATP. This is nothing but our 10th step of our glycolysis, which is resulting in formation of pyruvate. Okay, and this pyruvate is formed in the cytosol of the cell, but it has to be get converted into acetyl-CoA. And how is it possible? It is getting converted into acetyl-CoA due to the action of the PHD complex. This PHD complex is nothing but the pyruvate dehydrogenase complex. Okay, pyruvate dehydrogenase complex, which is located within the mitochondrial matrix. This is the mitochondrial matrix, and this PHD complex is located inside the mitochondrial matrix. This helps in oxidative decarboxylation of pyruvate to acetyl CoA. So this complex is decarboxylating our pyruvate into acetyl CoA inside the mitochondrial matrix. Okay, this is a large enzyme complex called as PHD complex, which is nothing but our pyruvate dehydrogenase complex. The pyruvate in presence of CoA and NAD is this NAD is reduced to NADH and is converted into acetyl CoA, where decarboxylation has occurred and CO2 is generated. This step is the most important step which links between the glycolysis and the TCA cycle. So it is linking our glycolysis where our end product is pyruvate and it is converting it into acetyl CoA inside the mitochondria, and this acetyl CoA enters our TCA cycle. Thus, this is one of the most important steps which is connecting both the cycles, our glycolysis cycle as well as our TCA cycle. So now let's understand how is this complex inhibited. That does it keeps on working continuously, or is there some inhibition? Of course, there is some inhibition on this complex. Let's understand what is the inhibition. Before we start about the inhibition, you should understand that uh, when the indicators of the energy rich state of a cell. So how do you understand that the cell is energy rich? You can understand that when NADH and ATPs are in higher concentration, the cell is in energy rich state. Okay, when NADH and ATP are in higher concentration, the cell is in energy rich state. When there is NAD plus or AMP, you have to understand that the cell is in energy deficit state. So when the let's consider the condition that the first condition that the NADH is high. That means that the concentration of NADH is high. That means the cell is in high energy rich state. Therefore, we doesn't need TCA cycle more. Therefore, it inhibits the complex, PDH complex. Okay, when the cell is in energy rich state, that acetyl CoA is also high, NADH is also high, ATP is also high. These are the indicators of the energy rich state. Therefore, they inhibit the PDH complex. Now, when the energy cell is in energy deficit state, we need TCA to run. Okay, we need TCA to begin. Therefore, what are the indicators of energy deficit state of the cell? NAD plus, that is oxidized form of NAD plus, CoA is rich and AMP is rich. Adenine monophosphate is high in concentration. NAD plus is high in concentration. CoA is high in concentration. Therefore, all three together, which are the indicators of energy deficit state of the cell, stimulates or triggers or ons the PDH complex so that it can convert the pyruvate into acetyl CoA and results in the higher concentration of acetyl CoA so that the TCA cycle begins. Therefore, this is an on and off mechanism of TCA cycle, one of the on and off mechanism of TCA cycle, where NADH, acetyl CoA and ATP, that is nothing but our energy rich state, inhibits the PDA complex, and NAD plus CoA and AMP stimulates or triggers the on of the TCA cycle. So students, now let's begin with the basic structure of TCA cycle. Here, this is a cyclic process. We have understood this is a cyclic process. Here, acetyl-CoA enters this TCA cycle, and acetyl-CoA is a two-carbon compound. So acetyl-CoA combines with the oxaloacetates and it gets converted into citrate, which is a six-carbon compound. Then citrate is getting converted into a isomer, that is isocitrate. This is also a six-carbon compound. Now, isocitrates is getting converted into alpha ketoglutarate, which is a five-carbon compound. So, what is happening over here? The six-carbon compound is getting converted into a five-carbon compound. This is because of the decarboxylation, which is happening over here. There is a generation of carbon dioxide. We have to understand this. When a six-carbon compound is getting reduced to a five-carbon compound, or a five-carbon compound is getting reduced into a four-carbon compound, there is a decarboxylation occurring over here. Therefore, alpha ketoglutarate is getting converted into succinyl-CoA, that is a four-carbon compound, and succinyl-CoA is getting converted into succinate, that is a four-carbon compound, and further conversion results into the fumarate. It is also a four-carbon compound, and fumarate is converted into malate, four-carbon compound, and further malate is converted into oxaloacetate. This is a generalized representation. We will deal with these steps in detail. Okay, I hope you are clear with this. There are two decarboxylation occurring over here. Okay, let's proceed ahead with this cycle. Students, now let's understand this TCA cycle in detail over here. This acetyl-CoA, if it has to enter into this TCA cycle, it must get cleaved. It must get cleaved into acetyl and its coenzyme A. So acetyl enters into the TCA cycle, and the CoA coenzyme A is cleaved out of the cycle, and the acetyl combines with oxaloacetate, forming an six-carbon compound citrate. The first product of TCA cycle is citrate. Therefore, it is also called as the citric acid cycle. Students, now let us understand what are the various byproducts that are released in this cycle. Here, the first is the addition of NADH. That NAD in its oxidized form is getting reduced into NADH. NADH is a highly energized molecule which is a precursor for various other biochemical cycles. So there are the three steps where we are getting three NADH molecules generated. When isocitrate is decarboxylated into alpha ketoglutarate, there is a generation of NADH. When alpha ketoglutarate is oxidatively decarbonized into succinyl-CoA, there is a generation of NADH. Similarly, when malate is converted into oxaloacetate, then there is a generation of NADH. As a process of decarboxylation, there is a resultant carbon dioxide is released. Okay, when one of the carbon is removed from isocitrate in the form of carbon dioxide, a five carbon compound alpha ketoglutarate is formed. Similarly, another carbon is removed in the form of carbon dioxide where succinyl CoA is formed. Okay, they are the two decarboxylation step which is occurring in this cycle. Further, we have generation of GTP, which is another energy-rich molecule. When succinyl-CoA is converted into succinate due to the action of succinyl synthase, there is a formation of GTP and FADH2. FADH2 is also an energy-rich compound which is generated during the conversion of succinate into fumarate. Thus, we have three NADH generated, one GTP generated, and FADH2 generated. These are the important byproducts of acetyl-CoA which makes this cycle energy yielding one. I hope you have understood till now. Now let's deal with the further aspects of this cycle. Here in this cycle, acetyl-CoA, before it enters citric acid cycle, it gets cleaved and the coenzyme CoA is released. Acetyl-CoA combines with oxaloacetate in presence of enzyme citrate synthase to form citrate. The citrate is converted into its isozyme isocitrate by the help of the enzyme aconitase. Now, the isocitrate, which is a 6-carbon molecule, gets decarbolized uh, to form alpha ketoglutarate in presence of the enzyme isocitrate dehydrogenase. As it is getting decarbonized, there is a result of CO2 and NADH, which is an energy-rich compound, which we have studied earlier in this site. Okay, in this slide. Okay, now when alpha ketoglutarate, a 5-carbon compound is formed, this is converted into succinyl CoA due to the action of the enzyme alpha ketoglutarate dehydrogenase. Here, oxidative decarboxylation is occurring, where again a NADH is generated and a carbon dioxide is released. But when there is a decarboxylation of alpha ketoglutarate is done, it combines with a coenzyme A to form succinyl CoA. Okay, this coenzyme enters the cycle over here and forms a succinyl CoA. Now, the succinyl CoA is getting converted into succinate by the action of enzyme called succinyl CoA synthase. Here, there is a substrate level phosphorylation occurring where GDP is converted into GTP. Thus, our GTP is another energy-rich molecule which is released over here. Now, the succinate, which is a 4-carbon compound, is cleaved from this CoA and the CoA leaves the cycle over here. This succinate is converted into fumarate by the action of enzyme succinate dehydrogenase. This is dehydrogenating the succinate into fumarate by a release of a of FADH2, which is another energy-rich molecule. This succinate dehydrogenase is very tightly associated in the matrix of the mitochondria. It is associated with complex 2 of an electron transport chain, which we will deal in our next class regarding this enzyme. Okay, once fumarate is generated, the fumarate is converted into malate by the enzyme fumarase, which hydrolyzes by the addition of water molecule to form malate. Malate now, in presence of malate dehydrogenase, undergoes dehydrogenation to form oxaloacetate. Thus, there is a resultant of NADH generation. There is no decarboxylation occurring here. Only dehydrogenation is happening. Therefore, only NADH is released over here. And once oxaloacetate is formed, the cycle again continues. I hope it is clear now. Regulation of TCA cycle. TCA is an energy-rich cycle, therefore it cannot be continuously on always. It needs to generate energy only in response to the energy needs of the cell. We had earlier read about the regulation of the PDH complex in response to the cellular energy needs, and there are 3 enzymes which can also be regulated within the TCA cycle. One is the citrate synthase, which combines oxaloacetate and acetyl-CoA. The second is an isocitrate dehydrogenase, which converts isocitrate into alpha ketoglutarate. Then the third one is alpha ketoglutarate dehydrogenase, which converts alpha glutarate into succinyl-CoA. These three enzymes can be regulated. Let's see how. Before I start this, students, I had told you earlier that ATP, that is adenosine triphosphate, and NADH and acetyl-CoA are the indicators of high energy in the cell. But whereas ADP, that is adenosine diphosphate or adenosine monophosphate, AMP, are the indications of the low energy of the cell. Therefore, when there is a low energy, the enzyme has to get activated. When there is a high energy, the enzymes must be inhibited. Therefore, here you can see that ATP, NADH, and acyl-CoA and succinyl-CoA inhibits the citrate synthesis, and ATP and NADH inhibits the isocitrate dehydrogenase. NADH and succinyl-CoA inhibits the alpha-ketoglutarate. Okay, and now when the condition of the energy states are low in the cell, which is indicated by ADP here, ADP and ADP, it is activating the isocitrate dehydrogenase. Okay. Therefore, availability of ADP is very important for TCA cycle to produce because it's activating the various enzymes. As ADP, that is adenosine diphosphate, is an indicator of the low energy of the cell. Thus, I can conclude that in general, the TCA cycle is inhibited by high energy charge, which is indicated by adenosine triphosphate and NADH, and stimulated by low energy charge, that is ADP or AMP molecules, which activates various enzymes. So students, here is an overview that we understood that glycolysis produces the pyruvate by the oxidation of glucose. This pyruvate is then oxidized to the acetyl-CoA in the mitochondria. The acetyl-CoA units are oxidized to carbon dioxide by the TCA cycle in the mitochondrial matrix. When you combine these two, glycolysis and TCA, there is energy generation in form of NADH molecules and FADH2 molecules and ATPs, where NADH and FADH2 are carried forward into the electron transport chain in the mitochondrial matrix. So the significance of TCA cycle is that there is a complete oxidation of acetyl-CoA into carbon dioxide and water. There is a generation of ATP, but in the form of indirectly in the form of NADH and FADH. This is in final oxidative pathway for various molecules like proteins, fats, and carbohydrates. Integration of major metabolic pathway. This cycle is interlinking different individual metabolic pathways. Okay, excess carbohydrates are converted as a neutral fat. There is no net synthesis of carbohydrates from fat. Carbon skeleton of amino acids, finally they also enter the TCA cycle. Therefore, the various products are entering the TCA cycle, and TCA is acting as a final common oxidative pathway. Students, we have to understand that there is no direct participation of oxygen in TCA cycle, but TCA cycle operates only under aerobic conditions. This is because that NAD plus and FAD plus required for the operation of this cycle can be generated in the respiratory chain only in presence of oxygen. Therefore, the citric acid cycle is strictly aerobic. So now let's discuss the energetics of the TCA cycle. A single molecule of acetyl-CoA, there are three NAD plus coenzyme included. We had studied this earlier at various steps, and one FAD molecule included and one GDP is included with addition of two water molecules. when fumarate is converted into malate and when oxaloacetate is converted into citrate, there is an addition of water molecules to water molecules. And now the resultant is 2 carbon dioxide. This is because of the decarboxylation occurring when isocitrate is converted into alpha ketoglutarate and the alpha ketoglutarate is converted into succinyl CoA. That time we have this carbon dioxide releases, okay, in these two steps. Then we have CoA releasing in the form of succinyl CoA to the succinate conversion. You have a CoA release, and then there is a 3 NADH generated in various steps, which I had discussed in the earlier slides, that 3 NADH molecules were generated and one FADH2 molecule is generated. One substrate level phosphorylation is occurring when succinyl CoA is converted into succinyl and in hydrogen. This is general energetics of TCA cycle. Thus, it can be concluded that oxidation of 3 NADH when it is coupled with electron transport chain, it results in the synthesis of 9 ATP. Therefore, 1 NADH results in the generation of 3 ATPs in electron transport chain. Okay. Therefore, when 3 NADH are involved, 9 ATPs are developed or produced in the electron transport chain. This electron transport chain will be dealt in our next class. Uh, FADH2 is comparatively less energy molecule when it compared to NADH. Therefore, it generates only 2 ATP in the electron transport chain. One substrate level phosphorylation is occurring, that is GDP is converted into GTP. Therefore, you can tell that total 12, 9 plus 2 plus 1. Therefore, total 12 ATPs are produced from one acetyl-CoA when it is combined with the electron transport chain. Okay. This is the energetics of an the TCA cycle. Thank you, students, for listening to my class. This concludes our TCA cycle. In next class, we will be dealing with an electron transport chain in detail. Thank you, one and all.